In this video, I will be explaining enzymes and how their activity can be influenced by inhibitor molecules. What are enzymes? Enzymes are special proteins that help speed up chemical reactions within living organisms. They work by binding to molecules called substrates at specific areas in their structures called the active site. Substrates can only bind to the active site. After the reaction is complete, the products are released and the enzyme can be used again. Enzymes are highly effective at speeding up chemical reactions, but enzyme activity can be affected by other molecules known as inhibitors. Inhibitors can slow down or even stop the chemical reaction, and as a result, slowing or preventing the production of products. Enzyme inhibitor molecules are generally classified into two main types: competitive and non-competitive inhibition. Starting with competitive inhibition, as the name suggests, the inhibitor molecule competes with the substrate for the binding to the enzyme's active site. These competitive inhibitors resemble the actual substrate structure, allowing them to fit into the active site. However, unlike the real substrate, the competitive inhibitor does not undergo a chemical reaction and is not converted into products. By occupying the active site, competitive inhibitors successfully block the substrate from binding, which reduces or completely stops the enzyme's activity. An important factor of competitive inhibition is that because the inhibitor is competitive with the substrate, as more substrate molecules come available, they are more likely to outcompete the inhibitor for the binding to the active site, restoring the enzyme's activity. A real-life example of a competitive inhibitor is the drug methotrexate. In the body, an enzyme called dihydrofolate reductase normally binds with folic acid to help produce nucleic acids, which are needed for DNA synthesis. Methotrexate is similar in structure to folic acid, so as it competes for the same active site on the enzyme. When methotrexate binds to the enzyme, it forms an enzyme inhibitor complex that stops the reaction from proceeding. This stops the production of nucleic acids, which slows down cell division, helping slow cancer growth. To better understand how competitive inhibition works, we could break it down into a simple analogy. Imagine you are trying to play and progress in a video game. You are the substrate, and the controller is the enzyme, and your sibling wants to play too, but you don't want them to play because they won't progress you in the game. They are the inhibitor. You both compete for the controller, just like the substrate and inhibitor compete for the binding of the enzyme's active site. Sometimes your sibling manages to grab the controller from you first. You now cannot play, and you cannot progress in the game. However, if you have a friend over to play the game, you and your friend have a better chance of grabbing the controller before your sibling does, and therefore you can make progress in the game. Now that competitive inhibition has been explained, let's move on to non-competitive inhibition. In non-competitive inhibition, the inhibitor molecule binds to a different site on the enzyme called the allosteric site rather than the active site. This binding to the allosteric site changes the shape of the enzyme, which in turn alters the shape of the active site so that the substrate will no longer be able to fit properly. As a result, the enzyme's activity is reduced or completely stopped. Unlike competitive inhibition, in non-competitive inhibition, adding more substrate molecules does not help to produce more product as the enzyme itself has been changed and cannot function properly. A real-life example of non-competitive inhibition is cyanide. Cyanide inhibits an enzyme called cytochrome C oxidase, which is part of the electron transport chain in the mitochondria where cells produce energy. Instead of binding to the enzyme's active site, cyanide instead binds to the allosteric site, changing the enzyme shape and preventing it from catalyzing the conversion from oxygen to water. This prevents the flow of electrons through the electron transport chain, which means no ATP is being produced, leading to cell death. To better understand how non-competitive inhibition works, let's continue with our video game analogy. You are still the substrate, and the controller is the enzyme. Just like before, you are still trying to make progress in the game, just like the substrate, creating product with an enzyme. But this time, your sibling doesn't compete with you for the controller, but instead, while you're trying to make progress in the game, they go to the console and they cut the wire. Even though you're still holding the controller no longer works, and the game cannot progress as the connection has been broken. In contrast to competitive inhibition, in non-competitive inhibition, Adding more substrate, such as a friend similar to you, will not give you a better chance of making progress in your game, as the controller still doesn't work. So the next time you are competing with your sibling for the game controller, just remember you are acting out a real biochemical process. Whether it's competitive or non-competitive inhibition, enzymes and inhibitors are playing their own game behind the scenes.